Imagine trying to understand, like, this massive, epic story, right? But you only get fragments. Maybe ripped pages, garbled sentences, whole chapters just
Mm.
missing. For decades, that's pretty much been the reality in human genomics. We had a draft, sure, but huge, really important parts of our DNA were like dark matter, unreadable, like those missing chapters in the book of life itself. But here's the thing, it's changing. A whole new chapter is unfolding, like right now. What if we told you that these amazing new sequencing technologies are finally letting us read that book? Not just sentences, but like whole paragraphs, entire pages even. That's what this deep dive is all about, long read sequencing. So yeah, today we're really digging into the world of long-read sequencing in human genomics. We've looked at the latest research, gone through the sources you sent, and basically our mission is to give you the shortcut, get you completely up to speed on this really cutting-edge stuff. For those of you deep in genetics, maybe prepping for a big meeting, or maybe you're just super curious, understanding long-read sequencing, it's not just important, it's becoming essential. It's changing everything, how we find genetic variation, how we diagnose diseases, even how we think about the genome itself. You'll get the inside scoop on why it's so revolutionary, what problems it fixes that short reads just couldn't touch, and, what it means for personalized medicine down the line.
Exactly. And throughout this deep dive, we'll kick things off by defining long-read sequencing clearly. Crucially, we'll compare it to the older short-read methods, just to really highlight why this shift is such a big deal. Then we'll get into the tech itself, the major players driving this revolution. We're talking PacBio, HiFi, and Oxford Nanopore. We'll unpack how they work, their sort of unique error patterns, and their really powerful applications.
Okay, and then from there, we pivot to the applications, right? The really game-changing stuff, like finding those structural variants that we're always hiding, building truly complete gapeless genomes, untangling complex diseases, even figuring out which variants are on which chromosome, that phasing piece. We'll touch on the bioinformatics too, the software making it happen. And yeah, definitely cover the challenges because it's not all solved yet. It's going to be a pretty comprehensive journey, so let's jump in.
Okay, so let's start right there. Why did we even need long-read sequencing? I mean, short-read sequencing was the workhorse for years, right? It totally changed the game, became the standard. But like we hinted, it had its weak spots, its Achilles heel, especially with something as messy as the human genome. Why couldn't it give us the full picture, that complete book?
Yeah, it really boils down to a fundamental limitation, you could say. Short-read sequencing, SRS. It just fundamentally struggles with certain things. Highly repetitive sequences, imagine reading the same short phrase over and over, trying to piece together where it fits in a huge book. It's tough. And also complex structural variants, these bigger rearrangements in the DNA, and regions that look really, really similar to each other. It's like trying to reassemble a shredded newspaper where tons of pieces are just identical small ads, or the rips are too tiny to even span a single headline. These limits meant big chunks of the genome, those dark matter regions, stayed murky. They were gaps in the map, or just ambiguous blobs.
So we weren't just missing like occasional typos. We were missing entire paragraphs, maybe whole chapters that were actually crucial for the story. And these weren't just, blank spaces, right? These regions actually do important things.
Oh, absolutely, far from it. These dark regions, they include critical stuff like centromeres, absolutely essential for chromosomes to divide properly. They've got tons of transposons, these jumping genes, and VNTRs, variable number tandem repeats, which change a lot between people. In those spots, short reads they just can't be placed uniquely or assembled correctly. They're too short, they don't span the whole repeat, or they cross the breakpoints of a big structural change. You lose the context. You don't know where that piece goes. And here's the really critical bit, the clinical impact. A lot of disease-causing variants, the pathogenic ones, they happen to be right in these difficult regions. So short reads made them invisible or super hard to interpret. We're talking big structural changes, inversions, gene copies, things with major health effects. Short reads just couldn't see them properly. Left us with these diagnostic blind spots.
Diagnostic blind spot.
But
Wow, that's huge. So, okay, how do we actually fix that? What are the technologies, the breakthroughs that let long-read sequencing see what short reads missed?
right, so that brings us to the main technologies making this happen today. There's PacBio, specifically their HiFi reads, and then there's Oxford Nanopore Technologies, or ONT. Why don't we start with PacBio, their SMRT sequencing single molecule real time.
PacBio, yeah, that name's definitely familiar. They've been around, pushing boundaries. But maybe refresh our memory a bit on the details. How does SMRT actually read those super long DNA pieces?
Okay, so PacBio's SMRT tech uses these really specialized chips, flow cells, with tiny, tiny wells, picoliter size. They call them ZMWs, zero mode waveguides. And at the bottom of each tiny well, there's a single polymerase enzyme stuck there. Then you take your DNA, circularize it, they call this a SMRT bell, kind of clever, and feed it into the well. As this SMRT well template goes through the polymerase, the enzyme adds bases, A, T, C, G, that are tagged with fluorescent colors. And here's the cool part. They watch this happen in real time, recording the flash of light from each base as it's added, right through the bottom of the well. So you're literally watching DNA synthesis happen, base by base.
Wow, okay. Super direct observation. But I do remember hearing, maybe early on, the PacBio reads had higher error rates. How did they get past that to these HiFi reads everyone talks about now?
You're right. Yeah, the early days were different. Initial SMRT reads, they were long-ish, maybe 1.5 kilobases average. But the raw error rate was higher, like 13%, mostly false insertions, adding extra bases. But, and this was the key discovery, those errors were randomly distributed, just scattered randomly along the read. So if you read the same molecule multiple times, the chances were that the random errors would pop up in different spots each time. And A might be misread as a T here on pass 1, but it'd likely be correct on pass 2.
Ah, okay. So the randomness itself became the solution. Just read it enough times and the errors cancel out. That's, yeah, that's ingenious.
Yeah.
How did that lead to HiFi?
Exactly. That insight led straight to CCS, circular consensus sequencing. If you have that circular SMRT build template, right, the polymerase just goes around and around reading the same molecule multiple times, making multiple passes. Then, computationally, you build a consensus sequence from all those passes. It averages out the base calls, effectively correcting those random errors. And if you get enough passes, say 10 or more, these consensus reads, the HiFi reads, achieve incredible accuracy, like 99.8% accuracy. That's Q30 quality. Q30, for anyone who tracks quality scores, means just one error in 1,000 bases. And crucially, they're still long, average 13.5 kilobases, often much longer.
Okay, so it really is the best of both worlds then. Long reads, but with that short read level accuracy, that must have huge implications. What can you actually do with reads like that?
Well, the accuracy means HiFi reads are now basically on par with short reads for finding small variants, like SNPs and small indels. Under 50 base pairs, we're talking over 99.9% precision and recall for SNPs, and over 95.9% for indels and structural variants, which is phenomenal. And that combination, the accuracy plus the length, is what lets you do really high-quality de novo genome assembly. Building a genome map from scratch. We're seeing contigent 50s. That's a measure of how long the assembled pieces are, over 15 megabases. And the accuracy is like 99.997% correct compared to known truth. That's just an amazing level of detail and completeness, especially for those really tough regions. It gets us way closer to truly gapeless genomes.
15 megabases. That's incredible continuity. So given that accuracy, what are the downsides, or maybe just the trade-offs with PacBio HiFi now? Is it cost, sample requirements?
Yeah, those are the main things. While the accuracy is fantastic, the cost per gigabase or per genome is still generally higher than bulk short-read sequencing, especially if you're doing huge population studies, thousands and thousands of samples. And the other thing is DNA input. PacBio HiFi generally works best with a decent amount of high-quality, high molecular weight DNA. So starting material that's really intact, if your sample is old or degraded or you only have a tiny bit, that can sometimes be a challenge.
Okay, that makes sense. So PacBio gets accuracy via that multi-pass consensus. But Oxford Nanopore, ONT, they're doing something totally different, reading electrical signals in real time. How does that work, and what does it mean for their capabilities?
Yeah, it's a completely different physical principle. ONT passes a single strand of DNA through a nanopore, a tiny protein channel embedded in a membrane. There's an enzyme, like a motor protein attached, which controls the speed, kind of ratcheting the DNA through the pore. And as each different base A, T, C, or G squeezes through that tiny opening, it disrupts the electrical current flowing across the membrane in a unique way. They measure these tiny changes in current, amplify them, and decode that signal in real time to figure out which base just went through.
So it's literally reading the electrical fingerprint of each base as it passes through this microscopic hole. Wild. And I hear about their ultra-long reads. Is there even a limit?
That's one of the most striking things about ONT. Theoretically, there's no upper limit to the read length, other than how long a piece of DNA you can actually get into the sequencer without breaking it. If you could isolate a 10 megabase molecule, ONT could potentially read it. In practice, they routinely get reads over 10 kilobases. And yeah, ultra-long reads over a megabase have been achieved, which is just huge for spanning complex regions. Throughput-wise, they scale up pretty well too. The Gridion platform can do, say, up to 100 gigabases in a run. The big Prometheon can hit up to 6 terabases. Plus, they have the portable Minion, that little handheld thing. That's opened up sequencing in completely new places, field labs, remote clinics. Incredible versatility.
Portability and ultra-long reads. Definitely unique advantages. But what about the accuracy side compared to HiFi? PacBio had random errors, but ONT's profile is different, right?
Or systematic. Yeah, the raw accuracy of ONT reads is typically lower than HiFi, more like those early PacBio reads. The main error types are false deletions, missing bases, and notably homopolymer errors. So inaccuracies in long stretches of the same base, like a brick hallway. And the key difference, as you said, is that unlike PacBio's mostly random errors that average out with more coverage, ONT errors can be more systematic. It's like the pore has a slight accent when reading certain sequences or lengths. So just reading it more times doesn't automatically fix everything. You need smarter bioinformatics, often using things like neural networks that are trained to recognize and correct those specific systematic error patterns. Coverage helps, but the algorithms are critical for high accuracy.
Got it. So different error types need different correction strategies, but ONT has another trick up its sleeve, right? Something about detecting modifications directly on the DNA.
Yes, this is a huge differentiator. ONT can sequence native DNA or RNA directly, without needing chemical treatments like bisulfite conversion for methylation, which can damage the DNA. It reads the molecule as it actually exists in the cell. This means it can detect epigenetic modifications like DNA methylation, 5-MeC, 5-ACMANC, 6-MeMe, or RNA modifications in real time at base pair resolution. And because it's reading one long molecule, it can phase these epigenetic marks with a genetic variance on the same strand. That's massive for understanding how genetics and epigenetics interact, especially in those messy, repetitive regions where epigenetics plays a big regulatory role. You get sequence and epigenetics in one shot.
Sequence and epigenetics. Wow, okay. And what's this read until thing I've heard about? It sounds like it chooses what to sequence.
Exactly. Because the sequencing is happening in real time, the software can actually analyze the first part of a DNA molecule as it starts going through the pore. And if it decides, based on that initial sequence, nope, not interested in this piece, it can actually reverse the voltage and eject that molecule from the pore. That's read until. It lets you do adaptive sampling, basically. Focus your sequencing power only on the regions you care about. It's been used really effectively for targeted sequencing, like creating a panel just for known disease-causing short tandem repeats, STRs. Instead of sequencing the whole genome, you just enrich for reads hitting those 37 or so critical STR genes involved in neurological diseases, saving time and money.
That is incredibly cool. Like a smart filter, just rejecting the junk and focusing on the important bits. You can see how that speeds things up for diagnostics, maybe for those STR expansion disorders like Huntington's, getting a quick, accurate count of those repeats. Okay, awesome. We've got the tech down. Now let's really talk impact. What have these long reads actually unlocked? Especially think about those structural variants, the SVs. We used to talk mostly about SNP, single base changes, but SVs, the big deletions, duplications, inversions, they affect way more DNA, right? But they were so hard to see properly before. How exactly are long reads a game changer here?
It's night and day, really. The ability of LRS to just span the entire structural variant, including its breakpoints and the surrounding sequence, that's the core advantage. Short reads would fall into the gaps or couldn't resolve the complexity. They were functionally blind to much of this. Just look at the numbers. One big study looked at 32 people from diverse backgrounds using LRS. They found over 107,000 SVs. But here's the mind-blowing part. Only about 30% less than a third of those SVs could even be detected using standard short-read data from the exact same people.
70% missed. Wow. That's not a blind spot. That's like a whole dimension of variation we were missing. What kinds of SVs were hiding in that 70%?
A really big chunk involves these things called transposable element-mediated rearrangements, or TEMRs. Basically, rearrangements caused by jumping genes. LRS has just blown the lid off how common and important these are. Alu elements, for example, these very common mobile elements in our genome, are responsible for maybe 80% of TEMRs. They often cause rearrangements through homologous recombination, but sometimes using surprisingly short stretches of similarity. These TEs aren't just hopping around inserting themselves, they're acting as breakpoints, as substrates for all sorts of larger rearrangements, creating something like 635 kilobases of structural changes in every genome on average. And it's often the younger TEs, the ones still active in humans, that are most involved. This matters because these TE-driven rearrangements often hit genes, impacting function, causing disease, driving evolution. It's a major source of variation we just couldn't see clearly before.
So these jumping genes are constantly reshuffling the deck, causing way more large-scale variation than we realized. Okay, what about inversions, flipping segments of DNA around? Those sound tough, too.
Yeah, inversions are super tricky for short reads. Why? Because usually no DNA is gained or lost, it's just flipped. So copy number looks normal. And the breakpoints where the flip happens are often in repetitive sequences. Short reads just get lost. LRS, though, especially with smart alignment software, dramatically improves detection. We can now see hundreds of inversions. Many of them happen again and again in the population, recurrent inversions, and they're often flanked by these long inverted segmental duplications, really similar chunks of DNA but facing opposite ways. That structure strongly suggests they happen through a process called NAHR, non-allelic homologous recombination. Just look at the Y chromosome. We now know it has recurrent inversions covering about 3.6 megabases. That's like 6% of its sequence. Details we just didn't have.
And what about genes where we have multiple copies? Or those complex duplications where copies are stuck together? Can long reads sort those out too?
Absolutely. Resolving complex tandem duplications and multi-copy CNVs or mCNVs is another big win for LRS. Short reads just can't figure out the exact number of copies or how they're arranged because the reads are shorter than the duplicated unit. LRS can span the whole thing, telling you exactly how many copies of a gene like GPRN2 or SPD-White-E2 are there and in what order. That's critical for understanding gene dosage, how much protein gets made, which can be really important for health and disease. Too many or too few copies can cause problems.
Right. Okay, so we've had the human genome reference, GRC38, for a while now, but we keep hearing it was more of a draft, especially in tricky spots. What's the impact of LRS on finally completing that map, getting a true telomere-to-telomere, end-to-end assembly? It feels like finally getting the blueprints for the whole building, not just most of it.
That's a great analogy. The development of truly complete, gapeless human genomes like the T2T CHM13 project is honestly a landmark achievement. And it was only possible because of long reads. It gives us this complete, uninterrupted view of parts of the genome that were previously just blanks or fuzzy, structurally weird regions. GRCH38, our workhorse reference, it had known gaps, big gaps, and poorly defined regions, especially in centromeres, telomeres, and the short arms of those acrocentric chromosomes. They were literally question marks on the map, or regions known to be wrong or incomplete.
And now with P2T, those question marks are filled in. What did we find hiding in those previously unmapped areas?
It's pretty staggering. The T2T assembly showed that over 81 megabases of sequence, mostly in segmental duplications, these long, nearly identical repeats were either totally missing from GRCH38 or represented incorrectly, with the wrong copy number or structure. That includes something like a 70% increase in the amount of highly identical segmental duplication pairs, over 95% identity. Short reads just collapsed these, couldn't tell them apart. Long reads resolved them beautifully.
81 megabases. That's huge. Is it just repetitive stuff, or does it contain important genes and variation?
Oh, it's definitely not just junk. These newly resolved regions are hotspots for human genetic variation. They contain variants in critical genes, genes involved in brain development like TBC1D3, disease-linked genes like LPA for heart disease risk, SMN for spinal muscular atrophy. It highlights just how important these complex regions are for understanding human diversity, disease, evolution. They weren't empty spaces. They were crucial, highly variable parts of the genome we just couldn't see properly.
Okay, that connects perfectly to haplotype phasing. Knowing if two variants are on the same copy of a chromosome in cis or on opposite copies in trans, that distinction can be critical, right? How have long reads made that easier?
Yeah, phasing is a classic problem where long reads offer a massive advantage. It's super important for diagnostics. Think about recessive diseases. You need two bad copies of a gene, one inherited from each parent. So you need to know if the two variants you found are in trans on different chromosomes that actually cause the disease. It's also key for figuring out if a new mutation, a de novo one, came from the mother's or father's side, which affects counseling for future pregnancies. Traditionally, short reads needed fancy statistics or family data or population frequencies to infer the phase. It was often indirect. Long reads, they are ideal for phasing because one single long read can physically span multiple variant sites. It directly shows you which variants travel together on that specific DNA molecule. Less need for parental samples, less reliance on statistical guesswork.
So you're not guessing anymore, you're actually seeing which variants are linked on the same strand. What tools help with this, and how good can it get?
WhatsApp is a really well-known tool for this. It's built for third-gen sequencing, PacBio and ONT. It does highly accurate read-based phasing. You can even integrate family info if you have it, making the phasing even more robust. And the ultra-long reads you get from ONT, they take this even further. They can phase enormous regions, like the entire major histocompatibility complex, the MAC region, which is incredibly complex and vital for immunity. Getting that phased correctly into one block, that was basically impossible before. Huge implications for immunology, autoimmune disease, transplant matching.
Okay, this really sounds like it's changing clinical practice. So bringing it back to patients, can long-read sequencing genuinely change a diagnosis today, make a real difference?
Without a doubt, LRS is moving rapidly into personalized medicine. We're already seeing major successes in neurology, cancer, inherited diseases. It's not just research anymore. It's finding pathogenic variants that were completely missed by standard tests, even whole exome sequencing or microarrays. These are answers patients couldn't get before.
Finding things missed by the standard tests. Are there studies showing this actually increases the number of diagnoses made, leading to better care?
Yes, definitely. Studies are clearly showing improved diagnostic yields with LRS, which translates to better patient management. For example, there was a case where LRS identified a specific deletion in the PCSK9 gene that's important for cholesterol levels. This deletion was hard to spot with other methods, but LRS found it, and it was linked to much lower LDL cholesterol. For someone with stubborn high cholesterol, knowing they have that specific variant could totally change their treatment plan.
Can you give us a few more concrete examples? Where is LRS really shining in specific disease areas?
Sure. Inherited metabolic disorders are a big one. LRS is great at sorting out complex gene duplications, deletions, rearrangements that cause conditions like Prader-Willi or Angelman syndromes or mitochondrial diseases. These often involve tricky genomic regions. In cancer, LRS is huge for finding fusion genes, where bits of different chromosomes get stuck together, or characterizing really complex events like chromothripsis, where chromosomes just shatter and get stitched back together randomly. Understanding these drivers is key for choosing targeted therapies.
So it's finding the exact broken parts to target treatment more effectively.
Exactly, and pharmacogenomics predicting drug response. LRS can fully resolve really complex genes like CYP2D6, which metabolizes tons of common drugs. These genes have lots of structural variation. Getting the sequence right with LRS gives a much better prediction of how a patient will handle certain medications, allowing for personalized dosing. And of course, for rare disease discovery, LRS is uncovering novel disease genes in large research consortia focused on unsolved cases. It's pushing the frontier.
It seems like it's impacting almost every corner of clinical genetics. What about bigger picture stuff, like newborn screening? Could we use this preventatively?
People are definitely looking into it. Pilot studies suggest it's feasible to integrate comprehensive LRS-based genomic analysis into newborn screening. It hints at a future where maybe we get a detailed genomic picture early on, identifying risks, guiding preventive care, maybe even before symptoms show up. And that targeted application we mentioned, using ONT's read until for those STR expansions. That's incredibly impactful right now for conditions like Huntington's, Fragile X, Friedreich's ataxia, getting an accurate count of those repeats of the diagnosis. LRS with read until gives you that precise count directly, much better than older methods like Southern blots. Faster, more definitive answers for families.
Okay, the power is undeniable, but turning all that raw, long-read data into actual answers, that requires some serious computational muscle and smart software, right?
Right.
What are the key bioinformatics tools and maybe emerging approaches that make sense of it all, taking us from just reads to, the full story?
Absolutely. The bioinformatics is just as crucial as the sequencing tech. For just aligning the reads back to a reference, tools like NGMLR, or the very popular Minimap2, are optimized for long reads. Then, for finding structural variants from those alignments, Sniffles is a big name. It's designed to handle the error profiles of long reads, using different signals to spot SVs. There are others too, like PBSV, SVIM, DELI. Often the best approach is actually an ensemble method. You run several different SV callers, maybe some designed for short reads too, like MANTA or LOMPY, plus the long read ones, and then you combine their results, apply filters, trying to get the most accurate, lowest false positive call set, especially for tricky things like those TE-mediated rearrangements.
So you combine different tool strengths to get the best overall picture. Make sense? What about building the whole genome from scratch or doing that phasing?
Right, for de novo assembly, especially aiming for that telomere to telomere quality, you've got tools like Canu, Falcon, WTDBG2, and newer ones like Genome or Hifiasm, which were often tuned for high-accuracy HiFi reads or specifically designed to handle those tough segmental duplications. And for phasing, yeah, WhatsApp is still a go-to tool for doing that highly accurate phasing based on the reads themselves, sometimes adding in family info for extra power.
Okay, and I keep hearing about pangenomes and graph genomics. How do long reads fit into that, moving beyond just one reference sequence?
Yeah, that's a really fundamental shift happening right now, and long reads are essential enablers. We have so many genomes sequenced now, hundreds of thousands. Relying on just one reference, like GRCH38, which is based on just a few people, doesn't capture all the diversity. It introduces bias.
Mm.
So the field is moving to graph-based pangenomes. Think of it like a map that includes all the different roads and detours found across many different individuals, not just one route. Tools like minigraph, PGGB, minigraph cactus are used to build these complex graph structures. The advantage? They represent all known variation much more efficiently. This improves read mapping, helps find complex SVs that might not even exist in the linear reference, and gives a richer view of diversity. And they're surprisingly compact. You can store billions of bases of sequence variation in just a few gigabytes.
Wow, a map of all human variation. That's powerful. Are there tools specifically designed to use these graph pangenomes now?
Yes, that's the next wave. We're seeing graph-aware tools emerge. Things like SFARP for finding SVs within the context of the graph, or Giggles for genotyping SVs using the graph information. There are also quality control tools like Flagger, which uses reads to check for potential misassemblies within these complex graph-based assemblies, ensuring they're accurate.
Cool. And beyond SVs and assembly, any other specialized long-read tools opening new doors?
Oh yeah, definitely. For detecting those base modifications with ONT, you have tools like Nanopolish, Signal Align, Deep Signal, Macaulay, Epinano. Many use sophisticated machine learning like hidden Markov models or neural nets to decode those subtle electrical signals into epigenetic information. And transcriptomics studying RNA is exploding with long-read tools. They finally let us see full-length RNA transcripts properly, which short reads struggle with. This gives much better insight into alternative splicing, different versions of genes, isoforms, and quantifying gene expression accurately. It's a really exciting time for bioinformatics innovation driven by this data.
It sounds incredibly promising, almost like we're right there about to have the complete instruction manual. But, there are always hurdles. What are the big challenges still facing long-read sequencing before it's just standard practice everywhere?
Yeah, despite all the progress, there are definitely still obstacles. A big one, honestly, is still cost. It's come down a lot, but LRS is generally still more expensive per sample than bulk short-read sequencing. For huge population studies or for routine clinical use across the board, that cost difference matters. The initial equipment cost is also higher.
Okay, so cost is still a factor limiting scale. What about the accuracy? You mentioned ONT errors are more systematic. Is getting perfect accuracy still a work in progress?
It is, yeah. Raw long reads, especially from ONT, do have higher error rates, and those tricky systematic errors like in homopolymers. Getting those absolutely right requires sophisticated correction, and it's not always perfect. PacBio HiFi is incredibly accurate, but maintaining that super high accuracy consistently across different sample types, chemistries, and at ever-increasing throughputs, that's an ongoing challenge for R&D. The bioinformatics keeps getting better, but raw error is still a factor to manage.
And the sheer amount of data, the computational side must be intense too.
Oh, absolutely. Analyzing LRS data is computationally heavy. The files are huge. Aligning them, calling complex SVs, assembling genomes, it takes serious computing power, powerful servers, lots of RAM, big storage, and specialized bioinformatics expertise. Not every lab or clinic has that readily available.
Making the analysis pipelines more user-friendly, maybe cloud-based, is really key to making the tech more accessible outside of specialized centers.
Right. Not just the processing, but just storing terabytes of data per project must be a headache. It really is. Data storage is a significant challenge. Even with clever compression, like in pangenome graphs, managing these massive data sets is tough. And then there's the final piece, clinical validation and functional interpretation. We have amazing examples, but we still need more large-scale studies systematically using LRS in big cohorts of patients with unsolved diseases compared to controls to really prove its clinical utility broadly. And often, even when LRS finds a compelling new variant, especially in SV, you still need follow-up experiments in cells or model organisms to functionally prove it's the cause of the disease. That step from correlation to causation is still critical.
Okay, that makes sense, but still, what's really striking here is that long-read sequencing isn't just a small step up. It feels like a fundamental shift. We're finally getting to look into these hidden corners of the genome, moving from that fragmented draft to something truly comprehensive, filling in those missing chapters.
It really is. And if you think about the future, where this is all heading, it's incredibly exciting. We are genuinely on the cusp of the next big sequencing revolution. That goal of routine whole genome sequencing using long reads, getting an individual, fully phased, de novo assembled genome for everyone. That's not science fiction anymore. It's becoming achievable. Imagine a single, truly generic genetic test that finds all types of variation, SNPs, indels, SVs, maybe even epigenetic marks all at once. That's the potential here.
A single test for everything, a complete personal genetic blueprint, phased end to end. Yeah, this is a profound vision for health.
Exactly. And think about real-time diagnostics, too. Those portable ONT devices. They open up possibilities for rapid diagnosis right at the point of care in clinics, maybe even tracking outbreaks in the field. We've seen proof of concept for things like identifying bacterial pathogens in hours in a hospital. All this comprehensive genomic data will fuel truly personalized medicine, guiding targeted therapies with much higher precision, predicting drug responses better, informing genetic counseling with way more detail. It enables a shift towards proactive, predictive healthcare.
And what about understanding us as a species, all of humanity? How does this complete map change that?
That's where those pangenome efforts are so important. The Human Pangenome Reference Consortium, HPRC, and similar projects globally. They're building these diverse graph-based references. They aim to capture the full spectrum of human genetic diversity, moving beyond a single reference that doesn't represent everyone. This will give us a much deeper understanding of population-specific variations, human evolution, migration patterns, a truly global, equitable view of our shared genetic heritage.
So wrapping up, what does this all mean for you listening and maybe for science itself when we can finally see every piece of the puzzle, resolve every SV, fill every gap, map all the diversity, what new diseases that we finally understand, what new treatments could emerge, and maybe how will this complete, unbiased map of human variation reshape how we even think about ourselves, about what it means to be human? It feels like the journey is really just beginning, and the insights from long-read sequencing are going to keep redefining health and disease for a long time to come.